Structure of proteins. Proteins are the polymers of alpha amino acids. Amino acids are joined by peptide bond or peptide linkage. Chemically, peptide linkage is an amide formed between COOH group and NH2 group with elimination of water. When carboxyl group of glycine combines with the amino group of alanine, we get a dipeptide known as glycylalanine. If another amino acid joins a dipeptide, then the product is called a tripeptide. A tripeptide contains 3 amino acids linked by 2 peptide linkages. Similarly, when 4, 5 or 6 amino acids are linked, the respective products are known as tetrapeptide, pentapeptide or hexapeptide. When the number of amino acids joined together is more than 10, then the products are called polypeptides. A polypeptide with more than 100 amino acid residues having molecular mass higher than 10,000 units is called a protein. However, the distinction between a polypeptide and a protein is not very sharp. Polypeptides with fewer amino acids are also called proteins if they have a well-defined conformation of a protein. Example, insulin, which has 51 amino acids. Let us understand the detailed structure of proteins. Structure and shape of proteins can be studied at 4 different levels, that is, primary, secondary, tertiary, and quaternary, each level being more complex than the previous one. Click on the buttons to learn more. Primary structure of proteins. Proteins may have one or more polypeptide chains. Polypeptide chain has amino acids linked with each other in a specific sequence, and this sequence of amino acids is called the primary structure of that protein. Any change in this primary structure, that is, the sequence of amino acids, creates a different protein. Secondary structure of proteins. The shape in which a long polypeptide chain can exist in secondary structure of the protein. The secondary structure could be alpha helix and beta pleated sheet. These structures arise due to the regular folding of the backbone of the polypeptide chain due to hydrogen bonding between the ketone group and NH groups of the peptide bond. Alpha helical structure. In alpha helix, a polypeptide chain forms all possible hydrogen bonds by twisting into a right-handed screw with the NH group of each amino acid residue hydrogen bonded to the C double bond O of an adjacent turn of the helix. Beta pleated sheet structure of proteins. In this structure, the peptide chains are stretched out to nearly maximum extension and then laid antiparallel to each other. Intermolecular hydrogen bonds hold the structure that is like pleated folds of drapery and therefore is known as beta pleated sheet. Tertiary structure of proteins. The tertiary structure of proteins represents overall folding of the polypeptide chains, that is, further folding of the secondary structure. Tertiary structure gives rise to two major molecular shapes, that is, fibrous and globular. Quaternary structure of proteins. The animation shows the quaternary structure of proteins. Some proteins are composed of two or more polypeptide chains referred to as subunits. The spatial arrangement of these subunits with respect to each other is known as the quaternary structure. A diagrammatic representation of all these four structures is given below, where each colored ball represents an amino acid. Protein's final structure is dependent upon the components of all four levels. Proteins can be classified into two types on the basis of their molecular shape. A. Fibrous proteins. When the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds, then fiber-like structure is formed. Such proteins are generally insoluble in water. The examples are keratin, which is present in hair, wool, silk; myosin, which is present in muscles. B. Globular proteins. They have chains of polypeptides coiled around to give a spherical shape. They are usually soluble in water. The examples are insulin and albumins.